Okay. Prepare. Okay. Yay, RuleBender. So Mary wants to switch to a different workspace. So I hope I have one handy. I think I do. So what you want to do, you want to go to the window menu in your RuleBender app and select switch workspace.
Oh, I see. Okay.
And then you can browse to the location where that is. So for me, it would be in my Dropbox, and I think, are you out there, Kunal? I think I have my student, what's going on here? Oh, it does not, it doesn't, it doesn't want me to pull that down for some reason to make it easier to find all the files.
Okay.
I'm just looking, so I mean, I can, oh, so like, let's just take a walk back into history. So I can pull up a workspace from a previous.
Well, I seem to be there.
And then, so you just hit, so you select that folder and then you hit OK. And then it gives you some, it asks you several times if you want to upgrade. And then it's just all, life goes on. Let's see.
Okay, so I just did that.
And then it should bring back up the new workspace. So here I have what we did in 2017.
Well, it restarted, but I still don't see anything in the navigator, which is odd.
But okay, but there were projects in the folder that you had before.
Yeah.
Yeah, that's a glitch of some kind. If it is the same folder, then it should see all the folders that are there.
Maybe I didn't select enough. Maybe I selected short of the folder. So let me try that again.
Yeah, it has to be the top level directory of that workspace, but
yeah, it says, well, browse. Oh, so maybe.
Gives me an opportunity to do something that I had wanted to do to start out.
Oh, that's annoying. No, it still doesn't go there.
Huh.
Or I don't see the files.
That is.
Damn it.
Not you, but there are folders in that directory.
Well, let me just check to make sure. Yes, there are.
Well, we'll try. I don't know. Yeah.
So should I select where it says RuleBender Workspace, or should I select where it says RuleBender Workspace CMW 2020?
You should select RuleBender Workspace.
Yeah, that's what I did.
Yeah, the top one, not the.
Right, what I did, and it didn't work.
If you want, we could ask. It'd be fun. Ron is asking if you want to meet with Sinan in a room, in a private room. It sounds so strange.
Well, I don't want to miss the presentation, so let's forget about it for now.
We're happy to help you get that sorted. Worst case is you have to regenerate that project in the BNGL file, but that file is available to copy and paste in pretty easily. So
where's it available to copy?
I will show you that in momentarily.
Okay.
So so we started out, and I think it's a good place to start out with RuleBender because the interface itself, especially for the people who are not used to working with an Eclipse or some kind of similar IDE, it has a lot of different functionality that is not easy to find by diffusive random walk, at least unless you're very adventurous. And you see that there are some pitfalls potholes you can fall into that are quite deep. So I think it's really good to do work with the interface and to show working with simple models. But of course, we didn't write RuleBender to do SIR models. It turns out to be good to, for quick and quick implementation and look and see what's happening in the model. It's great, but you know, we wrote BioNetGen to really handle complex molecular signaling networks. And actually, the sort of founding model, the model of these that we worked with in BioNetGen was this model of signaling downstream of this immunoreceptor called the high affinity receptor for IgE, also known as Fc epsilon R1. And the signaling involves the initial signaling events. I can't help wanting to rearrange the contact map to make it look prettier. The signaling events downstream of this receptor involve two tyrosine kinases, Lin and Syk, both of which can associate with different receptors of this multi-subunit receptor, which is labeled here as REC. And this is a funny contact map version. Anyway, and so it turns out that for a variety of reasons, basically having to do with the fact that the receptor itself, the ligand that binds to the receptor signals by forming receptor aggregates, and the simplest aggregate that can form is a dimer of receptors, which is sort of indicated here. The contact map is a little defective in that it doesn't show the link between one of the sites on the ligand and the receptor, but it does show the other one. It's just the quirk of the way it's set up. If we just move the sites over here, we can kind of obscure that omission. But so this ligand binds, it forms dimers of receptors, and then the receptor can get phosphorylated at both these beta and gamma subunits. And actually, I can show you the states. So there are phosphorylation sites on the receptor. There are phosphorylation sites on the tyrosine kinase Syk. There are actually phosphorylation sites on Lin as well that regulate its activity. They just weren't included in this original model. And what we found was when you make the rules that describe all the interactions, and all the rules are represented here by edges in the contact map or by components of these molecules that take on different states. So phosphorylation, in order to become phosphorylated, Syk has to undergo a phosphorylation reaction at these different sites. So there are a total of about 19, something on the order of 20 rules in this model. There are 19 in this version. Some of them are irreversible, some of them are reversible. And those 20 rules applied to the 4 original molecules in the system. There's the ligand, you can see them in the contact map, Lin, Syk, and receptor, generate something like more than 3,000 reactions. So if I just, let me see what's in this model before I blindly go running it. There's 2, oh yeah, I would say this is a fun simulation. So if I just say, okay, I'm gonna run this one, and we see that now in this console, it's actually the network generation part actually is involving some work. So this action generate network, it scrolls by, it's still pretty fast, this simulation. Yeah, so we got some interesting time courses, but let's go back to our console for a minute. It's not being cooperative, it's this one. So if we go back up here to the network generation, so we see that we started with our 4 species and no reactions. And as we started applying the rules, we generate more and more and more species and more reactions until at the end of the day, at iteration 11, we have a lot of species. We have 354 species and 3,680 reactions. So in order to simulate the dynamics of the system, we have to solve 354 differential equations that have a lot of terms. And I see people are asking the code. I'm going to show where some of these models are located. Most of the models that have been developed in BioNetGen and have been published are available somewhere online. We try to make sure that happens. I'm a little behind in going out and capturing some of the later models that have been developed, but usually the code is available. And one of the really nice advantages of the language is that the model files are very easily transferable. So yes, I'll show you where you can get this FC epsilon R1 example and ones that are more complicated. So at any rate, you see that, okay, we can generate the network and simulate it in a few seconds. And actually, most of the time, I shouldn't have done that. I should have held this up a bit longer for a sec. Come on, console. So most of the time that running the simulation took was actually generating this network, which is pretty big. And then it used to take minutes to do that, but computers have gotten faster. But the simulation time was a few seconds for these 300 equations. So it's solving the ODEs is pretty fast. Figuring out what the species are and doing all the bookkeeping is slower. Back in the beginning, when we first started developing these models, we actually tried to write out all of the differential equations by hand. And we actually had a committee. There were 6 of us, and we tried to do about 600, would have been 600 reactions each. I think we tried to do something like 60 equations each. And that proved to be quite difficult. Much better to make a computer program to generate those than to actually try to identify them by hand. One of the big problems is that a lot of the equations have factors of 2, either in the denominator and the numerator, and it gets pretty tricky to make sure you've got all those right. And of course, then if you were to change the logic in any way, then you have to go back to the drawing board and do the whole thing again. Whereas the computer is very happy to generate any network structure you give it. So so this has been really I think a useful tool. I'm just looking to see the time courses look like time courses of the kinds of species that we have in other kinds of models. And actually, this particular system in the configuration that we have with these parameters, it doesn't have a lot of complex dynamics sort of hardwired into it, but there is some subtlety here, and we learned a fair bit from studying this network. And I won't talk too much about the biology of it, but if people are interested, I can point you some papers. I think some of the things that we learned from these early efforts really had to do with how complex the structure of the complexes underneath the receptor activation were, and how that can have sort of a profound effect on the signal processing. And people are really still working out the implications of what it means to have to undergo a long sequence of reactions downstream of the receptor activation in order to activate the downstream molecules that ultimately are transcription factors. So let's see. Okay, so this is one model. So I wanted to impress upon you a couple of things. One is that we can do much more complex systems than SIR, and I think that if we went through each of these rules one by one, step by step, over the course of 6 hours or so, I think I'm not sure how much people would take away from it. But we can definitely go into more detail on some of these models, and I'll show some of the concepts over the course of this afternoon. The other thing I wanted to mention here was just this visualization of the network. So the complexity of the thousands of reactions and the hundreds of species is all very succinctly captured by this contact map view, which shows us most of the interactions, most of the relevant processes in the model. And you can actually browse this a bit more. The infamous the red line, which shows the interactions. It shows the binding interaction, for example, between the SH2 domain of the Lin tyrosine kinase and the phosphorylated beta subunit of the receptor. There's a trick here. So I have these upside down. The SH2 domain interacts with the phosphorylated beta subunit, but the unique domains appear. I'm messing up mine. The unique domain of Lin, sometimes called SH4, interacts with the unphosphorylated tyrosine of the beta subunit. It's the same motif in the beta subunit that it's a different tyrosine, I believe. But at any rate, there's actually much more complexity, obviously, underneath the signaling and the biochemistry than we've even represented here. And the original goal of BioNetGen was just to make it possible to represent that biochemistry in some compact way, and ultimately to be able to simulate the dynamics that's implied by that biochemistry. And what we'll find is that, you know, as powerful as the tools that we've been using so far are, they run out of steam at a certain level of complexity. So if we have too many tyrosine sites, if we have too many different protein binding partners, and too many different proteins in our system, we get so many species, so many differential equations that it's no longer really practical to solve them numerically, and we have to resort to some other kind of method for simulating the system, and that's where this so-called network-free methodology comes in, and I will at least give an overview of what that looks like today, you know, during the lecture part, and we may or may not get to an actual example of simulating that later on. Let's see. So people have been asking since I mentioned it. So if we go to the easiest way to show you how to get to where the models are is to go to the BioNetGen.org website. So all you really have to remember is BioNetGen.org, and then you can get to a directory, basically a menu that can take you to the relevant places. So I'm trying to remember. So the easiest place to find models, and some of the models we will be working with this afternoon, are in the BioNetGen tutorial on GitHub that's referenced here. So clicking on this link will take you to a repository in GitHub that's got some nice markdown description. So it takes you on an annotated guide through a bunch of tutorial models. I made that for the workshop a couple of years ago. And I quickly found out that I had way more models in that repository than I could possibly get through in a single day of teaching. But it's a really good resource for learning more. So that's a good place to go. I'll just mention that all of the code repositories for BioNetGen are on GitHub, and they're somewhat dramatically named the Rule World. The Rule World collection. There's a whole bunch of different tools here, some of which I'll be able to talk about, and others which I won't be able to talk about. But of particular interest to us is this BioNetGen, the BNG tutorial, which is up at the top. RuleBender and BioNetGen are the two main source code repositories. Atomizer is a tool that Sinan will be talking about later this afternoon. And one other area where we are trying to collect some of the models that have been published is in this supplemental materials repository that's unfortunately a bit underdeveloped. It's been neglected a bit in the last couple of years or so, but there are some nice models there still, and I'll go there first because there is a nice, I think, resource here. So there's an annotated table. This is a bit of an Easter egg. I see the way that it's labeled. But Saker 2017 actually has, it's best to go to the paper, I guess. But there, in this data set, S2 dataset.zip, not annotated, is a whole collection of models that we took from the literature and used applied visualization methods for. And there's like, so there's a whole bunch. There are like 28, I don't know, there's more than that. I think there's in the 30s at least models, including this the original IgE receptor model that I mentioned earlier. And I think also another very large scale model that I was going to show you from Helix in 2014. So yeah, they're out there. And like I said, if for some, it's not labeled the best the way that it is in this repo. But if you are lost and have trouble finding it, don't hesitate to just send a Slack message or whatever it takes to get our attention. So let's go back to, where was I in BioNetGen? I don't know what happened to my BioNetGen window, but okay, we'll go back here. This should have been, okay. So if we go back to the tutorial now, so this was another GitHub site. I lost, it was at the top of Rule World. So here you'll find a whole bunch of listings of models that are annotated with the language feature elements that they demonstrate. So a lot of the basic tutorial stuff that we did this morning is covered in slightly different form in this basic structure and syntax section. So we more or less went through the concepts that are in here, the parameter scan, all the basic model stuff, doing an SSA simulation. I used Latka-Volterra. The SIR model, a version of that is present here. Thought that was very appropriate for 2020. And so here's a version. This is the version where beta, where we didn't have R naught as the scan variable. But there's a number of other ones, and so like in the slides, it references Latka-Volterra, so there's an implementation of Latka-Volterra here. We've often used this as a demonstration. This is a very nice model to look at how it behaves in the BioNetGen context when you have the model as well mixed. So all of the species are uniformly distributed in space. There's a uniform concentration versus some interesting spatial effects that can arise in this system under certain circumstances when the reactions become, one or more of the reactions becomes diffusion limited. And this example exists as one of the demo examples in CellBlender. So either Tom may show it to you tomorrow, or you can certainly check it out once you start to get a working knowledge, know your way around the CellBlender interface. And when you do that, you can easily compare the model, the results that you get in CellBlender with the results you get in BioNetGen. And we will actually have a pipeline that we can, I'm not sure if Tom, are you planning to demonstrate the import, the CBNGL import tomorrow in CellBlender?
Yeah, I was going to show that, and I was wondering, what model you would recommend that we go back and forth with.
I mean, Latka-Volterra is the easiest one for sure.
Yeah, I think that'll be a good one to do. But it's not really a compartmental per se.
Yeah, there'll have to be. So I'll just walk you through a few. This will give you some ideas anyway.
Right. Okay.
So the tutorial is that we have the basic part, which we more or less covered this morning, and then the structured part, which we're going to cover some parts of this afternoon. I think you realize pretty quickly that we will not get through everything that's in here. There's really a lot of material and quite a few concepts. It always frustrates me that I can't show everything in one day, and I always have to remind myself, even if I could show everything, there's a limit to how much you can absorb inside of one day. At least for me, I know I would be pretty much done by some point not long after where we are right now. But the good thing is that all the models are available, all the resources are available. This time you now have a complete set of slides that walks you really through all of the stuff that's up here at the top. And you'll have slides for this part as well, for the structured molecules and rules. Not in the same level of detail quite, but pretty detailed. And they'll walk you through construction of a pretty complex rule-based model that shows binding and state change operations, which is what the focus is in these sections. There's some more kind of basic model kind of finishing the walk around of all the different capabilities. And then we get into starting to build towards more complex models of ligand receptor binding, and then and ultimately biochemical kinetics in the cell context. A good part of what's going on in the models that are in these sections are really building up a system of units that's systematic, that you can use for different models without getting really confused, which is very easy to do in the cell context. It's very easy to get confused about what's yeah, what the parameter issues are. We do not have mechanical forces explicitly implemented is one of the questions. That's a good, but the question is a good discussion topic for like a Thursday kind of session where we could talk about exactly how would you code a model in which the rate constants depended on some other characteristics. For example, they could depend on the concentrations of certain other molecules in the system could influence the rate constants in some ways. You could make a model in which accumulation of some product could affect essentially the kinetics of another reaction through an indirect mechanism. That's my best off the top of my head. I'm sure we can come up with better. Right, so there's more stuff down here. There's some cool visualization things, and I'm still going to. So what I wanted to get to was some of the larger models. So the question was asked, where did you get that FC epsilon R1 model? That one was. This is the original one from the JI paper from 2003. I think that's what I was showing you. If you just had this code without a nice contact map to guide you, it's a little bit of a daunting process to get involved in that, to get really understanding of what's going on in that model. There are quite a few other really cool models in here. The other one that I thought, let's see, I have the library. I don't have I'm just looking to see if I have another model. This model has essentially a number of the models in this section have essentially an infinite number of states. And so they can only be simulated. I hesitate to bring this into RuleBender because I don't know what it'll do to my computer. It's really, really humongous. This model has like 500 rules. It's actually fairly simple in its structure. It just has a lot of different of isoforms of the, not the right word, the subtypes of the epidermal growth factor receptor, so ERB1 through 4, and it has a bunch of different ligands in it, so it models all those combinations. And by the time you get done, this is several thousand lines of code to describe the model, but the number of implicit reactions generated by this, or species generated by this model, is something like 10 to the 12, and but we're able to simulate the model and rapidly enough that we can do parameter estimation on it using this NFSim tool. So I think I'm gonna not. I was thinking about bringing up a different model of the T-cell receptor signaling system, but it doesn't seem to be in this suite. So I will defer that to a little bit later. But the point is that you can, the tools are really meant to be scalable to large systems. And although it's hard to, fully walk someone through one of these detailed models in a relatively short session, and maybe not that useful, when you're just learning the tool, there's really a lot to explore here and a lot of capability. So I hope that's helpful. Diffusion limited model, yes. I think Tom will tell you, will give you, and working with CellBlender will give you some nice ways to understand what I mean when I say that a reaction is diffusion limited, because you'll come across some examples of that tomorrow, I believe.
Yes, the concept of diffusion limited versus reaction rate limited doesn't really mean anything at all in a well-mixed model where you don't really model the process of diffusion. It means nothing at all. And so you'll get to see tomorrow what what can go on in a spatial model when you have something that really is diffusion limited. And we'll play with the Latka-Volterra model a little bit to demonstrate the concepts there.
Yeah. Yeah, Latka-Volterra has some really bizarre behavior in the diffusion limited regime. It basically means that the reactions, so the core concept is that, in order for two things to react, they have to come close to each other. And if the reactions are very fast, then all the things that are near each other get depleted quickly, and what the time that it takes for another reaction to occur is waiting for molecules to get close to each other again. So
right.
Yeah, I see. I neglected to update the title slide of this presentation, even though I did update some of the slides, modify some of the slides I'm using from the presentation I gave last year. So don't worry that it's not like I'm just reusing the slides from last year and I put no effort in. I put effort in. Okay. So the key idea I just wanted to illustrate here, and as I've been mentioning, is this idea that we're looking for scalable methods to model cell regulatory networks. And these networks have potentially, and in real cells take lots of different inputs. They integrate them together in complex ways using very complex, detailed biochemical signaling pathways. And most of the models that we use to describe the kinetics of these systems abstract away a large amount of that detail. And that's all well and good, and we need to make abstractions to make successful models. But there are always cases where we don't know what the effect of making those approximations is. We may want to consider some interactions in greater detail, perhaps because there's a drug candidate that could affect the system at that level of detail, and we don't know how that interaction couples into this larger network. Then it's very hard to predict the effect. So there's always a need for greater detail in the description of cell signaling models. And ultimately, our vision in developing tools like BioNetGen was to be able to model the complexity at arbitrarily or detailed, high resolution, and be able to zoom out back and forth between high resolution and and a more coarse-grained resolution. Because whether or not it ever becomes possible to simulate the whole cell at sort of atomistic levels of detail, there are real questions about whether it's ever worthwhile to do such a thing. But there's a lot that we don't know about the effects of the approximations that we typically make. So it's good to be able to make realistic models. We can always make them simpler, but you can't take a simple model and identify whether the approximations that it makes are correct without making the more detailed model, if that makes sense. So ultimately, what we typically do when we study signal transduction, we study these pathways sort of with one spotlight at a time, usually. One part of the cell, one part of the cell signaling network, one subset of the inputs, and one subset of the outputs that can be measured experimentally. And ultimately, though, we're interested in how all of these work together. And we need models to be able to do that, because the wiring underneath is extremely complex. Not only is there this issue of sort of combinatorial explosion of complexes that we'll talk about going forward, but there's also as we'll see as well, feedback loops that make the dynamics even of very simplified circuits extremely complicated, and then finding those feedbacks in this very rich structured molecular environment is not always straightforward. So one of the driving motivations, as I mentioned here, is this idea of an integrated cell model. People have really started to do this in the last 4 or 5 years. Put together a bunch of different modules, signaling processes, cell cycle regulation, DNA damage repair, transcription translation, put them all together into an integrated model that can predict the dynamics of cell behavior. And it's real question of how to do that in a way that scales with all of these complex working parts. And the machinery of the reaction network that I showed in, that I started with can be scaled up to quite an impressive degree, but there are lots of kinds of areas where it does not scale well, and particularly anytime there are multivalent interactions, the number of different species that you have to track in such a network model becomes very large. And so what you wind up doing is saying, well, we'll model this protein which has 15 different phosphorylation sites, each of which is governed by different upstream kinases, and each of which regulates different downstream processes. We'll just lump them all together, and that's not very practical, and it has a lot of potential problems. So again, we're trying to make large-scale models based on large-scale maps. There's as complicated as this is, it hides an enormous degree of complexity, and it's not clear that at this level of abstraction the models are really predictive. So another problem that really comes about too is that when the models are coded in a language, a basic level computer language like MATLAB or C, a lot of models are coded in C. Everyone codes them in a somewhat different way, and the meaning of the code is very difficult to interpret. And anybody who's tried to look at somebody else's large-scale model that was coded in MATLAB understands exactly the difficulties that gives rise to. And having a structured language that's restricted, that's really meant to describe the basic level of interactions, makes the model much more easily understood. And that's that's the goal of like showing you a picture of the contact map, that underlies this enormous, complex reaction network with hundreds of species. So, okay, so going into some of the sort of more practical ground level view, how do we translate intracellular biochemistry into what we call executable models? So models that we can run simulations of a computer, analyze their dynamics, and hopefully learn something new. And so I'm listing, okay, some of the different challenges, and I alluded to these already. The idea that a protein is just a simple node in a network is something that anybody who studies proteins knows is not true. CAM kinase 2 is an incredibly complicated machine. It has an enormous number of different states and functions that are highly contextually depend on the proteins that are surrounding it, depend on its phosphorylation state, depend on the concentrations of a whole bunch of other regulators, ions, and so forth. And just thinking about protein tyrosine kinase signaling, which is where we started, proteins have multiple sites of phosphorylation, tyrosine, and also serine and threonine, and those phosphorylation sites mediate interactions with other signaling proteins that have highly conserved domains that recognize those modifications. And phosphorylation is just one of the modifications that takes place in signal transduction. Ubiquitination being another major one that's quite understudied compared to phosphorylation, in part because of the lack of really good antibodies. And a lot of what we know about protein biochemistry as well is limited by the tools that we have to measure their states, which are growing but still limited, and there's a lot more to learn. Okay, so there's a lot of complexity that we can sort of scratch the surface of, and that's enough to tell us that we have a problem in being able to represent the complexity of the molecular interactions that arises just from what we know about biochemistry. And that's epitomized by this FC-epsilon-R1 network that I showed you earlier, where we have a small number of components, just the initiating kinases of this signaling system. And really, this is highly simplified. There are multiple regulatory steps, even upstream of, for example, the activation of Lin tyrosine kinase that are not included in our model. And we can describe those interactions in the biochemistry that takes place, in a fairly concise way using rules. And these are represented as cartoons in this diagram. They more or less correspond directly to the actual rules that are implemented in the model. But then when we apply these rules to these components, we find out that there's this hidden complexity, that there's an enormous number of different states of the complexes underneath that you know, are possible just given those interactions. So for the FC epsilon R1 signaling system, where you have dimers and where you have essentially three different modification states, the sites of this, we model it as a heterotrimeric receptor, but it's actually heterotetrameric. The gamma subunit is a dimer. It has two sites that could potentially bind the Syk tyrosine kinase. So the combinatorial complexity here is really a gross underestimation of the complexity that's underlying this system. So at the end, you may have to squint to see this on your screen, but there are 354. I already gave that number before. Species, and if we just consider trimerization of the receptor, which is actually, the receptor is actually thought to be active in larger aggregates, there's even more. And so this idea of the reaction network model that I described earlier is really coming under strain because of the multivalent interactions here. So this idea that there's a small number of components and interactions, and it gives rise to a huge number of possible species and reactions, that's this combinatorial complexity that I've talked about. And another example that I actually showed you this model, we scrolled through it. This was the Creamer model from 2012. And it's a model of epidermal growth factor receptor signaling. It has even more conserved tyrosines. There are 4 different types of receptor in this ERB family. And the complexity is just astronomical for all the different kinds of ways that these things can form. Now, of course, all of the complexes aren't necessarily important. We don't need to model, you know, each aggregation state in all of its gory detail, but coming up with ways to manage that multivalency and to get at the fact that the protein, that a receptor aggregate can be basically concurrently signaling in multiple different pathways, that is an important property that we're aiming to capture in models. So we can easily write down a model that describes the interactions that are taking place in these systems. And the problem is that it's very hard to simulate even based on an automatic expansion of the reaction network, such as I described that, and we walked through a little bit this morning with BioNetGen. So rule-based modeling is really an attempt to refine this idea of the reaction network modeling that we talked about this morning. And the real difference is that instead of having species that just represent indistinguishable pools of things, of individuals, our individuals have structure. So they have sites that can bind things, they have sites that can undergo post-translational modifications, they have components or attributes that can represent other complex properties. So these multifunctional enzymes that I mentioned before, like the Lin tyrosine kinase, it is a molecule, and we call these molecule types in the BioNetGen lingo. We'll make a molecule types block to describe each of the different proteins that we're going to model. It can have multiple functional elements that we choose out of the possible set of all the different elements that a protein could have. There's always more than we model, to be sure. So we choose those based on the biochemistry that we're interested in capturing in a particular model. So in this very ambitious model of Lin tyrosine kinase, we could include multiple SH domains, SH3 and SH2, a kinase domain, and various tyrosines that are known to modulate the activities of Lin. Both this is, I guess, the kinase activation. We phosphorylate tyrosine, and this is the so-called C-terminal tail of Lin tyrosine kinase that, when it's phosphorylated, causes the Lin to adopt an inhibitory state. It's SH2 domain actually binds the phosphorylated tyrosine 508. So in BioNetGen lingo, we call these components. In other rule-based lingos, they're sometimes called sites. But each molecule object is comprised of multiple components that themselves can interact with other components and can also have states. And these states are represented here. This is literally the syntax of a molecule type definition. It says that lin has binding sites, SH3, SH2, kine, and then it also has modification sites, Y397 and Y508, that can be either 0, represents unphosphorylated, or P phosphorylated. These, the tyrosine sites, any site can also, any component can also participate in a bond. And we don't have to declare in the type definition what molecules it can bind to. That comes later. Actually, that's defined by the rules. So we see how the complex signaling proteins can be abstracted, but in a more structured form than with a typical species, in this way, by having components. And we can see immediately that whereas a lin species could represent one protein and possibly one state of one protein, the lin molecule can be in a whole host of different states. Each of these sites could be bound or unbound. These sites can be phosphorylated, unphosphorylated. They can also be bound or unbound. So there may be really an exponential, 2 to the 3 to the 4 to the n, where n is the number of these sites, number of states of the lin molecule in a complex. And so that gives us a much richer representation of what we had before. So this multi-subunit receptor can be described in another abstract way. There are multiple ways to do this, actually. This is really the simplest, sort of most coarse-grained way, where each of the subunits is represented as a different complex, as a different component. And within each subunit, we have just one element of that subunit is represented as a state, is represented with state attributes. So here we have the beta subunit of the FC_1. It's going to bind the lin tyrosine kinase. It can also be phosphorylated or unphosphorylated, and that's with reference to a particular tyrosine site in the beta subunit. In the yes is the answer to the question that just came up. The gamma subunit can be phosphorylated or unphosphorylated. If you know about anything about these proteins, this is again a gross under approximation of the complexity that's there. Each ITAM has a minimum of 2 conserved tyrosines. They can be phosphorylated independently, so you can have singly and doubly phosphorylated states. The singly and doubly phosphorylated states bind with different affinities to the SH2 domaining proteins in different proteins, depending on that. So there's an enormous complexity that we've only really scratched the surface of, and that is really part of the regulatory dance that's part of the regulatory mechanisms that these receptors are involved in, and which in many cases we're still working to get more elucidation of. So the first part of the rule-based language is having structured molecules, structured objects, as opposed to monolithic objects. And the second part is to have rules that operate on those structured objects. So rules define the interactions of molecules, very simple rules. And what they do, so many rules are simple. They can be also very complicated, but they enable one to represent in a very concise way and precise way what we know about the biochemistry of the protein-protein interactions. So for example, we can take a piece of information like the lin SH2 domain binds to phosphorylated tyrosine on the beta, tyrosine 218 on the beta subunit of FC_R1, and we can write a rule that describes that very compactly. So this is, the same notation that we used before, except now the species that are reacting, and we're going to refer to these as reactant patterns. The patterns contain structured molecules. So here the SH2 domain is a component that's present, and the beta tyrosine 218 is present, and it must be phosphorylated for this reaction to take place. And then when this reaction takes place, it creates a bond. And the bond is the thing that's referred to by the exclamation mark. The exclamation mark is followed by an index that indicates the starting point of the bond referencing that index. Question? Sorry, someone just unmuted. So here, there's a bond. I think I have the, yeah, I've got some annotation here. So left-hand side is reactants, right-hand side is products. I don't need to tell you about this because we spent all morning looking at reactions. But these reactions are a little bit different in that they resolve the structure of the underlying molecular constituents. So here we're going to make explicitly a bond between Lin and the receptor. And the notation takes a little bit of getting used to, this idea, but of this exclamation mark and a link. But you can think of this as just like the opening of a bracket and the closing of a bracket, and the bracket is numbered 1. So that just references that these 2 points are connected. The 1 has no other meaning, so we could label this bond 0 or 5 or 10 or 100, whatever. The number is not important. It just, they have to match in order to create a bond between those 2 sites. So in a complete bond, you have to specify both ends of the bond. There have to be 2 unique places, 2 unique components that are decorated with this exclamation mark index. Yeah, and so what the question was asked, can you nest bonds? And the answer in current BioNetGen language, it's really just the 2 levels, and the components are the only thing that can be explicitly connected between bonds, by bonds. There are some languages out there that allow bonding between different levels, between, say, a molecule and a component, or you could have nested levels of molecules. As far as I know currently, I only know of one language in which that is, that simulations are allowed with that kind of nesting. And I think one reason it's not more commonly done is that it makes the simulation algorithms much more complex. And so it's hard to make efficient simulators with multiple levels of nesting. NFSim is a very efficient rule-based, and we'll see this network-free based simulation method. And it works on the 2-level concept, and sort of refactoring that is been something that we have not managed to get to do yet. But yeah, okay. It's always hard to know. I could talk on about that at length, but I think that's not the most germane point at the moment. So rate laws. We already talked about rate laws before. The default assumption is mass action kinetics. These represent constants in this case, although they can equally well represent functions. In the BioNetGen context, we can make the rate laws arbitrarily complex. Okay, so I'm gonna take a sip because my voice. That's the hardest thing about Zoom. You talk and talk and talk. Zoom lecture, you talk and talk and talk, and my voice wears out. I found that when I taught in front of a classroom, because I write on the board usually, my voice is fine for 80 minutes. But when I talk on Zoom for 80 minutes, ooh. Okay, so this is key. It's good to pause here, because this is really, if you understand this statement, don't write, don't care, then that's really 90% of rule-based modeling. So, right. So good questions that are coming up. And it's perfect. I love the feedback. I love feedback on Zoom too. I personally much prefer to write questions by typing than by asking. I get really stage fright when I have to ask questions in front of a large group. And I like to be able to type. So great to have all the questions. I will explain the dot in a moment. but let me just explain this don't write, don't care for a sec. So the key idea, the key principle of rule-based modeling is really that the reactions represent, the way that we're going to interpret the rule represents the most parsimonious, simplistic view of that reaction. I'm not sure if that's really the best way to say it, but that's a way to say it. The point being that whatever we don't write in the rule, and remember when we defined Lin and when we defined the receptor, they have other components, but we haven't mentioned them in the rule. And that means that they, according to this rule, they don't affect the ability of that rule to generate a reaction in the first part. So in this part of the rule defines, the reactants define what participants are eligible. The products define essentially how do we modify the participants to get to products. So here, the way that we modified them was to create a bond between two of the components that were involved, that were part of the constituent molecules. And then the rate law can be complex, but usually, again, it's mass action. So it's just KPL times the concentration of the species that, the reactant species that matched Lin and the reactant species that matched this receptor. And a couple of more words about what these patterns mean. So when we refer to a component in a reaction rule, the way that we interpret that is that this component must be bound. If we don't refer to a binding state, if there's no bond listed, and we're talking about a reactant, it has no, this means that this component is not bound to another component. So the rule literally says a free SH2 site on Lin is required to form a bond with a free beta tyrosine 218 on FcεR1 that's phosphorylated. And then we can form this bond. If the bond, if SH2 is bound to something else, then it doesn't apply as a reactant here. If this beta subunit is bound to something else, then this FcεR1 doesn't apply. On the flip side of that is that, do I have examples? No, I don't on this slide. We'll get to it a little bit later on. The flip side of that is that if, you know, whatever is happening at SH3 domain, whatever is happening with the kinase domain, whatever is happening with the tyrosine tail, whatever, none of that is going to affect the ability of this reaction to modify that Lin molecule. So this don't write, don't care is part of what gives you, what makes rule-based modeling a very concise way to describe a system. It's not making any additional assumptions about what properties the reactants must have. So each reaction can generate a large number of different reactions. Each rule can generate a large number of different reactions involving many different species. So hopefully will start to make some sense. We'll give some more examples of that going forward. The question about this dot, the dot just represents that these two molecules are part of the same complex. It's a, there are arguments about the dot. In this case, it's redundant because these two things are bound together. You could say that, well, of course, Lin and FcεR1 are in the same complex, but we can write rules in which the two molecules are joined together by the dot, but we don't say how they're bound. So we don't make an explicit bond between them, but they still have to be part of the same complex in order for that rule to apply. This creates some mischief, and you know, there's some technical aspects of that we won't get into now, but that's what it means. Right. Let's see. So, and then there's mass action kinetics, which we talked about before, and which applies. So if we generate an individual reaction based on this rule, we'll get a reaction of, this is the concentration of, yeah, the way this is written is a little bit, is slightly misleading. I'm not going to get into the details of that at the moment, but basically the total rate of all the reactions generated by this rule is given by the rate constant times the concentration of all the Lins that have a free SH2 domain summed up, and all the FcεR1s that have a free beta Y 218, sum of those, and that's the total rate. But in reality, normally that's not how we do the simulations. We actually generate specific reactions. So how does that work? So here's a model. This is a slightly, you know, a representation of the building blocks of that. Yeah, I'll answer that question in just a sec. The building blocks of FC epsilon R1 model that I described before, these four different molecule types, and that's described pictorially here, and then a listing of the rules. So we saw that in the model file that I showed, we had the molecule types block, and we had the 19 reaction rules that were described in the BioNetGen language. The question was asked, do we have to use, can we use concentrations, or should we use molecule counts? Pretty much always recommend to use molecule counts in these models because it enables you to do a stochastic simulation quite easily. But there's no requirement. You can work in whatever units of concentration are convenient. So you could do a simulation in nanomolar or micromolar. I usually recommend against molar units, and I'll just throw this out here as a random aside, but it's a bad idea to do cell simulations in molar units because you sometimes get, it causes havoc with some of the integrators. They don't handle the numerical underflow very well sometimes. So if all of your concentrations are less than 10 to the minus 8, sometimes you get very bad results. And I'm not sure, yeah. I'm not sure exactly, there's no simple way to fix that other than to use units in which the concentrations don't give underflow problems. Yeah, it's numerical stability for sure. Yeah, it annoys me that that's a problem, but it is a problem. Okay, so what happens is we take our 4 molecule types, we take our 19 rules, and I showed you this process. You iteratively apply the rules to the original, really it's the original species. You'll see that the molecule types, we pick basically the bare molecules as our initial species for network generation and then generate the network from those. So here, actually you can see it, here the 4 molecule types that are the 4 molecules that are used as the seed, these are the only things that have initially non-zero concentration when we set up the model, at least to equilibrate, are the ligand, lin, SYK, and receptor, and all of those are in what I like to call their ground state, so they're unbound and they're unphosphorylated typically. Those of you who have good eyes will notice that the notation has unfortunately changed slightly between the different slides. Of course, within a model, those would have to be consistent, but I guess I took these slides from slightly different places. So, right, we apply network generation, we generate lots of species, lots of reactions. They're all really boring. Some of them have factors of 2. Yeah, you don't really want to have to decipher what all of this means. You would prefer to really just to work at this sort of level of detail, maybe at this level of detail to write the rules has to be done, but you know, getting into the details of implications of every single reaction rule applied to every single species is definitely very tedious. Why do some species have 0? So what happens is, and Tom alluded to this earlier, you start with the only species that you have to seed in your model are the ones that initially have non-zero concentration. So if we just assume that you have a cell that's completely at rest, that there's no activation of receptors, and no activation of lin or SYK or ligand, and those of you who are sophisticated about the biochemistry know that this is not a realistic underlying assumption. But nonetheless, we can start there for the purposes of defining the network system. Then what we're essentially doing with this network generation process is identifying all the possible species that could ever be populated by the reaction rules that we've defined. So it defines all the possible configurations that can arise based on the reaction rules that we gave. And so it's possible for, you know, for example, the ligand to bind to the receptor, forms a ligand receptor complex. That ligand receptor complex can recruit lin. Lin can phosphorylate a receptor. Well, first the receptors have to form a dimer, but then when there's a dimer, the lin can transphosphorylate, and so on and so on and so on and so on until you get 354 different species. But all the species that are generated from this network generation initially have no concentration. They're not present in the system. They only form after reactions start to take place. So hopefully that explains why most of the species that we generate are not initially populated, and then it's only over time when you stimulate the system that most of them become populated. Now, of course, the cell, phosphorylation and dephosphorylation happen in a resting cell. The cell has what's called a basal state, so many of the reactions can take place even when there's no ligand present. And so often we have to do a process where we generate the network, say just from our 4 species, but then we allow the network to equilibrate first before we add ligand, so that we get the correct resting state of a cell in which there is phosphorylation and dephosphorylation taking place. In this system, it's actually the one constitutive reaction that occurs in this system is that Lin can bind to the receptor constitutively. The Lin receptor interaction doesn't require phosphorylation to take place, but the Lin that binds that receptor doesn't have a substrate to phosphorylate, because we assume that Lin can only phosphorylate receptors that are in complex, that are not the original receptor that Lin is bound to. And there were some old biochemical experiments that our original collaborator in this work, Henry Metzger, late Henry Metzger, did that seemed to really support that mechanistic hypothesis, and so we built it into the model. In fact, the Lin transphosphorylation assumption is a major part of the reason that we actually wrote something as complicated as BioNetGen to do this work, because if you don't have that, if you don't require trans phosphorylation, then it's much easier the network becomes much simpler. Okay, so I've kind of covered these things. I'm gonna hopefully be able to speed up. The goal is to get you done. We should probably try to finish this part in a half an hour. That gives, we're supposed to take a break too. I don't know what time are we due for a break? I can't, the 3 hours really gets to me.
I think we're supposed to take a break pretty soon, but
5:30.
5:30. Thank you.
Yeah,
In my time
7
zone.
minutes.
In 5 minutes, I have to take a break.
7 minutes.
I feel like I've been talking a lot, so it's probably a good thing. These kind of slides are in a way a summary of what I've talked about. Hopefully, kind of reinforce the ideas. We have these components. They have they can represent different aspects of these molecules. We have states that represent their different modifications and binding. And then we can have complexes, and we've seen an example of a complex. We had a different one. We had Lin and receptor, right? I think there are a bunch of different examples of complexes, and we have this kind of funny bond notation. Bonds can occur within a single molecule. That's this C-terminal autoinhibition. And so, right, we have this richness of what the language can describe, and then we have rules that talk about how bonds form and break up, and how states get changed. So, since the last part of the practical is really all about just making simple rules to describe binding, unbinding, and component state change, this is a good place to kind of to wrap up this component, and that is. How we write a rule that makes a bond. I referred to this already. We can have, we need to refer to component sites, to components rather, that are unbound, and then we create bonds between them. And this is an interesting reaction because it describes an IgE that has 2 free sites, 2 unbound A components, and one of them gets bound by the rule. And I saw that Mary had her hand up.
Yeah, I just was wondering about the terminology. Is the terminology change you referred to between U and O when you have U and UP or OP?
Oh, the terminology for phosphorylation, there's not a requirement. There's
It could
not,
be either.
usually we use zero. Usually these days, the standard that we've tried to at least implement for ourselves is 0 and P. But that I think in my slides, I have many different iterations of nomenclature that we used. And the LIN has a domain called
Yeah,
the U
that's
domain.
right. That's what I was going to just ask about that. So
It doesn't matter. I mean, the component states and the components live in different namespaces, as do and the U domain on LIN has nothing to do with the U state of the beta subunit of FC epsilon R1.
So does the tilde denote a state?
This tilde denotes state, yes.
Okay.
Yeah, that's pretty much it.
Thanks.
So all the nomenclature that 90% of what you need to know is that exclamation mark represents a bond. When you have a bond and you have multiple components, you do need a dot to join them. And the other thing is a component state is represented by a tilde. You can have a discrete set of states that each component can take. Usually it's binary, but it can be any number. And there just have to be, a discrete number of them. We don't have finite state attributes, continuous state attributes. And then you can have reactions that, rules that change a component state or create a bond. Those are sort of the two elemental kinds of rules. You can also, the reverse reaction obviously deletes this bond. So bond breaking is another main aspect. You can combine multiple operations in a single rule, but generally when we're trying to describe mechanistic systems, we try to avoid doing that, with the exception of catalyzed transformation. Sometimes we'll have a catalytic reaction, the bond will break and the product will form in the same, the product modification, substrate modification will occur in the same step. That's our final example model. Right. Another thing to keep in mind here, there's a question on the chat about the ordering of these things. These are graphs. The underlying representation here is that of a graph. So the order really doesn't matter for determining the identity of the graph. The only place where the ordering can really matter is that, like in this case of the FC epsilon R1, we have two copies of a molecule in the same rule. So if you change, but in this particular case, changing the ordering on the product side would probably not have an effect. But there are cases, the way that BioNetGen maps going from reactants to products does retain some memory of the order. So there are situations in which writing the molecules in a different order can affect the behavior. I think that is true of components, but those are even harder to construct. The short description would be to say that the ordering matters only in rare circumstances when you're doing pretty complicated things. Right. I think there's some more terminology here. And the most important thing is something that I've alluded to but haven't really shown you, is this idea of a pattern. And the patterns, the idea that the patterns select the properties of the molecules, but they don't care about the things that aren't mentioned in the pattern. So a really nice example is this receptor. It has to have a free A state, and it matches the receptor with its free A state, and it doesn't care about the states of beta and gamma. So we can have receptors that are phosphorylated or not. The binding is gonna apply equally to receptors that have been phosphorylated or not. That can have some interesting subtle effects in some of these systems. Right, and this was the key idea, and this is now repetition, that because you can match many different species, each rule can generate a large number of different reactions potentially. We're not going to do any model building examples of that with our remaining hands-on work today, but there's ample examples of that in the tutorial set that I showed, and particularly the FC epsilon R1 model. I would encourage you to look at, and if you're curious, how does this really work? the original paper, the JI paper that the reference is in the somewhere. It's either in the model file or it should be somewhere on the website pretty close at hand. It goes through all the gory details of where all the species come from, species and reactions. Once upon a time, I worked all of that out. So I think we'll skip the rest of this terminology, and I will